Chapter 16 takes us down to the molecular level for looking at models of inheritance. Looking at the DNA molecule itself, we've talked about the particulate nature of genes in Mendel's experiments. We talked about chromosomes, the chromosomal basis of inheritance. And now we're going to go down to the finer scale and look at the DNA molecule itself. And here we've got the famous scientists Watson and Crick, who won a Nobel Prize for resolving the structure of the DNA molecule, which led the way to understanding how it can encode the hereditary information that Mendel and Morgan studied. So, in 1953, Francis Crick and Jim Watson introduced a model that showed the structure of DNA as a double helix. We now know that DNA is the substance of inheritance that holds all of the hereditary information. It is encoded in all cells of our body, and DNA directs the development of traits that are biochemical, anatomical, physiological, and to a limited extent, behavioral as well. So, Thomas Hunt Morgan's lab showed that genes are located on chromosomes, but it still was not clear where that information was encoded. We knew that DNA and protein were both present in chromosomes, but which one was the genetic material? Was it the protein or was it the DNA? And for a while, the idea that protein was actually the molecule that held the genetic information was prevalent because protein is so diverse. There's 20 different amino acids. It made sense that protein should encode for information, whereas DNA, there's only 4 nucleotides. You know, it's like, which is a more expressive alphabet? An alphabet that has 4 letters or an alphabet that has 20 letters? As it turns out, DNA is the molecule that we're interested in now. And how this was discovered was found by studying organisms like pathogenic bacteria and viruses and other organisms. So in 1928, there was a scientist named Frederick Griffith who worked with 2 strains of a particular bacterium. If you have more than one bacterium, you've got bacteria. So one of these strains was pathogenic, caused disease, and the other one did not. And he called them the S strain and the R strain. So S was for smooth, and R was for rough. Because when he looked at these colonies growing on a Petri plate, he found that the pathogenic ones looked smooth around their edges, while the non-pathogenic ones looked rough around the edges. And the reason for that has to do with the ability of that bacterium to produce a capsule. You might remember a capsule is that kind of gooey substance that helps bacteria to adhere to their hosts and enables them to induce disease. So these R cells did not have a capsule, so it wasn't very easy for them to get a foothold inside the host body. And the mouse's immune system, when he injected the mouse with the rough cells, could fend off that infection. But the smooth cells that had the capsule, he could inject those into the mice, and sad for the mice, but fortunately for the bacteria, the mouse died. So in his experiments, he took living cells of the S strain as a control, inject them into the mice die. He took living R cells, no capsule, inject them into the mice. The mouse survives. He could take heat-killed S cells. So these were cells that were killed by heat. Still had the capsule, though. He could inject them into the mice, and they did not cause disease. But when he injected them with a mixture of living R cells and heat-killed S cells, he injected that cocktail into the mice, and they died. And when he went to re-isolate bacteria from the mice post-mortem, after they had died, he got living S cells. Which was weird because he didn't inject them with living S cells. He had the heat-killed S cells and the living R cells. So what he concluded was that there had to be some factor that could be, some chemical factor that could be transmitted from these dead S cells that could get into the R cells that could somehow train them to produce the capsule. Fast forward to 1944, Avery, McCarty, and McLeod announced that this transforming substance is in fact DNA. Because only the DNA, when they gave the DNA fraction to these harmless bacteria, could they produce a capsule and turn into pathogenic bacteria. If they gave them protein, it didn't work. If they gave them other macromolecules, they did not get this transforming power. But scientists were still skeptical because we didn't know that much about DNA, and it still seemed like 20 different amino acids is a better way to code for things than having only 4 nucleotides. So further evidence came to us from looking at bacteriophages, which are a type of virus that infect bacteria. Viruses, there are viruses that can pretty much infect all different kinds of living organisms, not just people, cats, dogs, plants, and bacteria can have viruses that will infect them. So here was the bacteriophage which demonstrated that DNA was actually what was able to transform bacteria. So viruses typically are very simple, do not have organelles, they're not cells, but they have some type of nucleic acid, and then they have a protein coat, but only the DNA gets injected into the cells. We know this now. So the experiments that were done by Hershey and Chase were kind of clever. So recall, protein has sulfhydryl groups in it on a couple of the amino acids. On cysteine and methionine, they've got sulfur in them. So they grew up bacteria with bacteriophage in the presence of radioactive sulfur. So they could track this radioactive label, and because they knew that the protein would become radioactive. They also used a similar system, but they used radioactive phosphorus. Now, there's no phosphorus in protein, but there is phosphorus in that sugar phosphate backbone of DNA. Okay, so they had 2 parallel experiments. One where they had radio-labeled sulfur, making radio-labeled protein, and one where they had radio-labeled phosphorus, producing radio-labeled DNA. So what they found was when they looked for the presence of the label in infected bacteria, they only found it in the bacteria where the radioactive phosphorus was used. So the protein was clearly not getting into the cells. It was only in the DNA that was getting into the cells. And this demonstrated that it wasn't the protein that was the portion with the transforming power. It was, in fact, the DNA. So, following up on these results, it was demonstrated that DNA is a polymer of nucleotides, and we know that nucleotides have nitrogenous bases, as well as these pentose 5-carbon sugars and a phosphate group. And then in 1950, another biochemist named Erwin Chargaff reported an interesting finding, which was that DNA composition varies between different types of organisms, between different species. Now, what's the significance of that? Well, we know that different organisms are encoded differently. You don't use the same information to produce a bacterium as you do a simple worm or a fish or an oxen. So it makes sense that whatever this set of instructions is should vary between different organisms. And if DNA was just there acting as a scaffold or a silent partner or wasn't really involved in serving as the molecule of heredity, it should be the same for all organisms. So Chargaff performed an experiment. He found that the proportions of the nucleotides were different when he looked at E. coli, a bacterium, wheat, which is a plant, sea urchins, salmon, humans, and oxen, which are all different types of animals. So not only did he find that these ratios varied, he also found that the proportion of adenine was usually very close to the proportion of thymine, and the same for guanine and cytosine. Now, it looks like they're all right around 25% in E. coli. But then if you look at wheat, it's about 28% adenine, close to 28% thymine, and about 22% guanine, 22% cytosine. Okay, so those are still relatively close, but sea urchin, 33% adenine, and about 32% thymine, about 17% guanine, 17% cytosine. Now we're starting to see a pattern developing here. Okay, salmon, about 30% adenine, 30% thymine, 20% guanine, 20% cytosine, and so on and so forth. So this led to Chargaff's rules. One is that the base composition of DNA is variable between species. So the ratios of A's, T's, C's, and G's in you and I is different from what might be found in a sea urchin or a bacterium or a plant. The other of Chargaff's rules is that in any given species, the number of A's and T's are roughly equal, and the number of G's and C's are roughly equal. So now we're getting a little bit closer to understanding how and why DNA was the molecule that is going to transmit hereditary information. So now we understand, thanks to all of these discoveries, what DNA looks like. We talked a little bit about this back in Chapter 5, and now we're back to talk about it in gory detail here. Now we remember from Chapter 5 that we've got our nitrogenous bases, which can either have one ring or two rings. That's to say we can have pyrimidines, long molecules with a meaning a small, long word meaning small molecule, or purines, small word meaning big molecule. So either one ring or two rings, and those are our nitrogenous bases. Those are connected to our 5-carbon sugar. For DNA, we know it's deoxyribose because it doesn't have oxygen here. 1, 2, 3, 4, 5 sugar carbons on the sugar. attached to each other by a sugar phosphate backbone. So the phosphate group attached to the 3' carbon on one sugar molecule is attached to the 5' carbon on the next molecule.